Hey everyone, and welcome back to our channel. Today, we re-diving deep inside the cell to explore one of its most fascinating and hard-working organelles, the Golgi Complex. You might also know it as the Golgi Apparatus or the Golgi body. It's like the cell's very own post office and packaging center, and without it, our cells would be in total chaos. So, who do we have to thank for discovering this amazing structure? That would be the Italian biologist Camillo Golgi, who first identified it way back in 1898. He found this intricate system in pretty much all eukaryotic cells, from the smallest yeast to the cells in our own bodies. It's a fundamental part of complex life. Let's talk about what it looks like. Imagine a stack of slightly curved, flattened pancakes. In cell biology, we call these flattened sacs cisternae. This stack of cisternae is the core of the Golgi complex, but it's not just a simple stack. It's a dynamic, bustling hub. Surrounding this central stack are tons of tiny bubbles called vesicles. Think of them as the mail trucks and delivery vans of the cell. The whole thing is a complex network of interconnected tubules and sacs, all working together. Now, this stack of pancakes has two distinct sides, or faces. One side is called the cis face, which is the receiving end. It's kind of convex, or curved outwards. This is where new cisternae are constantly being formed. How vesicles, those little bubbles we mentioned, pinch off from the smooth endoplasmic reticulum, or smoother, and travel to the Golgi. They fuse together at the cis face, adding a new layer to the stack. So the Golgi is always growing on one side. The other side is the transface, which is the shipping end. This face is concave or curved inwards. Here, the opposite process happens. The cisternae break up, pinching off to form new vesicles that are ready to be sent out to other parts of the cell or even outside the cell entirely. It's a continuous flow, like a factory assembly line. Materials come in one end, get processed in the middle, and get shipped out the other end. So, what exactly is the Golgi doing? What's its main job? Its most important function is processing and packaging cellular products, especially proteins. This is why cells that do a lot of secreting, like the cells in your pancreas that make insulin, are absolutely packed with Golgi complexes. They are secretion superstars. Here's how it works. Proteins are first made on the rough endoplasmic reticulum, the roux. From there, they travel through the smooth and are then sent over to the Golgi complex in transport vesicles. Once they arrive at the Golgi cis face, their journey through the stack begins. As they move from one cisterna to the next, they get modified. The Golgi can add sugars to them, fold them into their final shape, or cut them into smaller, active pieces. It's like quality control and customization all in one. After these proteins are fully processed and modified, they reach the transface. Here, the Golgi sorts them based on their final destination and packages them into new vesicles. These vesicles then bud off and act as delivery trucks. Some might travel to the cell membrane to release their contents outside the cell. This is called secretion. Others might be destined for other organelles within the cell. Speaking of other organelles, the Golgi is also a factory for creating them. Important organelles, like lysosomes, which are the cell's recycling centers, as well as peroxisomes and glyoxysomes, all originate from the Golgi complex. It packages the necessary enzymes and membranes and sends them off to do their jobs. The Golgi's role doesn't stop there. It's also involved in creating complex molecules called conjugated molecules. For example, when it adds a carbohydrate chain to a protein, it creates a glycoprotein. When it attaches a lipid to a protein, it forms a lipoprotein. These molecules are crucial for things like cell signaling and forming the cell membrane. And what about plant cells? The Golgi has a super important, unique job here, especially during cell division. When a plant cell divides, it needs to build a new cell wall between the two new daughter cells. Guess who provides the building materials? The Golgi Complex. It produces vesicles filled with all the necessary components for the cell wall. During the final stage of cell division, cytokinesis, these Golgi vesicles line up at the cell's equator. They then fuse together, forming a structure called the fragnoplast. This fragnoplast is the foundation from which the new, rigid cell wall is built. So, from discovering it over a century ago to understanding its many complex roles today, the Golgi complex has proven to be an indispensable part of the cell. It's the master sorter, modifier, and shipper that keeps the entire cellular city running smoothly. Thanks so much for watching. If you found this journey into the cell interesting, don't forget to hit that like button and subscribe for more biology adventures. See you in the next video.